Good afternoon, everyone, and welcome to the Allen School Colloquium. I'm Professor Jeffrey Hare, and I have the honor today of introducing our speaker, Fritz Lechschuss. So Fritz is a PhD candidate in computer science at Harvard University, where he works on scalable visual exploration systems for analyzing patterns in biomedical data, where he's advised by Hans-Peter Pfister. And so prior to his PhD, Fritz completed his bachelor's and master's degree in bioinformatics at the Freie Universität Berlin, and then spent a year as a visiting researcher at Harvard Medical School in the Department of Biomedical Informatics. And though originally rooted in bioinformatics context, Fritz has also made multiple core contributions to the field of data visualization and visual analysis. These include new methods for making sense of large collections of statistical graphics and novel tools for combining visualization and interactive machine learning to analyze sequential, such as time series data. And so in addition to elegantly integrating visualization, domain science, applied machine learning, and design, Fritz's work has been recognized with several awards. These include a Siebel Scholar Award, the Best Paper Award from Euroviz 2020, and a Best Paper Honorable Mention from IEEE InfoViz 2020. Now, please join me in welcoming Fritz Lechschuss.
All right. Hi, everyone, and thank you so much for inviting me. Super excited for the opportunity. And so this talk is all about interactive visual pattern exploration and how we can apply it at and across scales, given increasingly large and complex data. And let me point up front that all the projects I will present to you today are really the result of fruitful collaborations that I was fortunate to have over the last years. Now, before I dive into the details, I want to briefly put my research into the broader context of data analysis that goes all the way from collecting data, over gaining insights, to generating hypotheses, and then ultimately deriving knowledge. And so within this process, my work primarily targets this first step here from data to insights, which often involves a great deal of exploration, in particular visual exploration. And the ability to explore data visually is really critical for discovering and interpreting patterns that might otherwise go unnoticed. So for instance, plotting a numerical vector as a bar chart, like the data we see here, allows us to quickly find the minimum and maximum value and to get a good sense of the overall distribution. However, as the data increases in size and complexity, this kind of visual exploration can become challenging, even if the encoding is generally effective, as it is, for instance, in this case. And so, for instance, here in this larger bar chart, finding the largest value is already harder because one of the three bars that here stand out is located far from the others, which makes direct visual comparison much more involved. And things can get even more challenging if we want to compare many instances at once. And so here in this example, while it's relatively easy to parse an individual bar chart, it can be time consuming to find, for instance, outliers or cluster these collection of bar charts based on their pattern type. And so in the presence of more and more data, scalability really becomes a pervasive challenge in many different domains. And here are some real-world examples, including, for instance, time series, microscopy, and geospatial data shown here on the left, or large multi-scale network data drawn as a matrix here in the middle, as well as high number of pattern annotations or highly repeated measurements shown here on the right. And for effective pattern exploration at and across scales, we have to overcome the following challenges. So first of all, with larger and higher resolution data, we typically also have to deal with higher rendering times. But more importantly, how can we explore large gigabyte size data collaboratively over the web, where, like sending gigabyte size data back and forth is simply not feasible. A more fundamental challenge is the available screen space for visualizing data is ultimately limited by the display technology and human vision. So drawing more information or simply increasing the display size does not necessarily lead to more insights. And finally, in many scientific domains, data sets contain reoccurring patterns, or sometimes called motifs. And in that case, insights are typically drawn from comparing multiple instances rather than just looking at a single pattern. And for large data sets, these pattern instances can appear in the tens to hundreds of thousands while being sparsely distributed. And so faced with these challenges, in my research, I really focused on scalable data exploration, using visualization as the primary means to interact with data and human-centered machine learning to guide this exploration at scale. And within the scope, I focused on three key tasks, which are browsing, comparing, and finding patterns. And now, just to clarify, when I talk about patterns in the context of large data, I really mean any kind of like frequently reoccurring visual artifact. And that can, for instance, be like a car in a large gigapixel image, could also be a spike or a peak in a time series data set, or like this kind of dot-like pattern here in a matrix. Another reason why I conduct research in this area is because we are faced with all these scalability challenges when it comes to exploring genomes. And so let me give you a very high-level overview. So think of the genome as a linear sequence of roughly 3.1 billion characters called the DNA. So these A, C, Gs, and Ts here, which encode information for the main building blocks of our cells. But unfortunately, knowing just the sequence doesn't tell us how cells really work. So another important aspect to genomes are so-called DNA modifications, which kind of regulate which genes are actually being expressed and how much. And moreover, recent advances in biology have found that the spatial organization, so how the DNA folds, has also important implications on several genetic processes. And now biologists have developed a number of methods to capture these events. And really the takeaway from the slide here is that in order to understand how genomes work, we have to assess multiple measurements that capture different events across different levels of scale. And specifically, my PhD projects were part of a large initiative by NIH called the 4D Nucleome Project, which focused on genome folding. And so let me show you how this data actually looks like. So when the DNA string that we see here gets tightly packed to fit inside the cell nucleus, it has been shown to form physical interactions between two or more locations, as highlighted here in red and blue. And now, using a novel method, biologists are able to count these pairwise contacts between up to 30 million regions across the entire genome, which then ultimately leads to so-called genome interaction matrices that are typically visualized as a heat map, where in this case here, for instance, a dark cell indicates that two parts on the DNA have been in close contact frequently. And now, if we would want to visualize this huge matrix in its entirety using a high DPI monitor, we'd need a display panel that's roughly the size of 3.4 kilometers in width and height, which is of course infeasible. And so, following my quest for scalable data exploration, I have developed several new visualization systems and interaction techniques that make this exploration more efficient. And today I'll show you 4 projects that can be roughly sorted by their support for browsing, comparing, and finding patterns. First, I'll show you HiGlass, which is a platform for multi-scale browsing of genome interaction matrices, as well as other types of multi-scale visualizations. Then I'll introduce you to a technique called scalable insets, which we developed in order to make navigation in these multi-scale visualizations more efficient. For comparison of thousands of pattern instances, I'll then present you with a new exploration approach that we call interactive piling. And finally, I'll show you a system that we call Peaks, which enables analysts to build pattern classifiers interactively using visual feedback. And so, okay, no questions so far. If anyone has a question, like, just put it in the chat, and then before other breakpoints, I'll get. But so far we haven't any, so let's jump right into the first project. So kind of as a first step towards analyzing these large genome interaction matrices that I've shown you before, we've developed this tool called HiGlass. And we published HiGLAS in Genome Biology back in 2018, and it was later also featured in a blog post on Nature as one of the important tools for really making sense of these genome folding data. And so this is how HiGLAS looks like. HiGLAS is a web application for fast and continuous rendering, navigating and also comparing genome interaction matrices. And you can kind of think of it conceptually as being, a Google Maps interface to these matrices. And so you can freely pan and zoom, and you can also interactively adjust the visual encoding as we see here. But now a distinguishing features to other tools in the field is that HiGLAS allows us to interactively compose so-called linked views. And so for instance, here we're duplicating the view, and then on the right side, I'll now replace the existing matrix from a benign cell line and to contrast it with a cell line, with a cancer cell line, basically. And using this, this approach in HiGLAS, you can really quickly build sophisticated views for studying specific locations on the genome in great detail. But of course, genomics is not the only domain which has large multi-scale data. And so later on, we generalized this HiGLAS system to support other quantitative data types. And we also integrated it into the Project Jupyter environment, which we then presented at the SciPy conference in 2019. And the important aspect here is really that this integration and generalization allows us to stay closer to the data analysis environment, which makes it useful to test hypotheses more quickly. And so for instance, here in this example, we plot hourly temperature data from the last 65 years in 3 different cities as these 3 line charts here. And now we can directly within Jupyter Lab, you can explore this data interactively in the very same way I've just shown you before with these matrices. And so now HiGLAS supports a variety of different data types, as we can see here. But what's really exciting to see is that HiGLAS has made a strong impact in genomics. So our public instance alone, which you can reach at higlas.io, is used almost 2,000 times per month by more than 45 research labs across the world. But more importantly, HiGLAS really became the underlying platform that enabled us to develop and test new visualization and interaction techniques for exploring this kind of large scale and data. And so this really allowed us to build several of the follow-up tools that I'll show you. But coming back to the example from earlier, so here we see this contrast between no pattern on the left side and this distinct pattern here on the right side. But one aspect I haven't actually told you about is how I found this specific region in the genome. And as a matter of fact, I just browsed these 2 maps until I saw an interesting difference between the 2. But you can already imagine that is not efficient. And it's really becoming an issue when these patterns are so small and sparsely distributed throughout the map that you don't see them when you're zoomed out. And so to address this kind of navigational problem, we have developed a visualization technique for pattern-driven navigation in these multi-scale visualizations that we call scalable insets. And we published this work in at the IEEE InfoVis conference in 2019. And again, genome interaction matrices are certainly not the only type of multi-scale visualizations. So for instance, geographic maps and gigapixel images share very similar properties. And in all these cases here, exploring such multi-scale visualizations can be time-consuming because small patterns are not visible when we're zoomed out. And as we zoom in, as we see here, context is kind of lost. And so again, in this context, a local or small pattern can be, for instance, a park in a geographic map, or car as we've seen before, or a dot in a matrix visualization. And now in the following, I'll use the world map example here to illustrate the challenges in exploring such local patterns. And so let us imagine that COVID isn't a thing anymore for a moment, and we're trying to find a good ski area for our next vacation. And to make an informed decision now, we typically need two things. We need to know where ski areas are located and how they compare in terms of, for instance, the number and variety of slopes that they offer. Now, browsing the entire US West Coast here on the world map doesn't tell us which of these regions might be interesting to explore in details. And so, of course, we can zoom far into the map, and that will reveal the visual details about a particular ski area, like the one we're looking at here. But it doesn't allow us to directly compare the ski area that we just looked at with others that are far apart. Now, assuming we have some means of extracting all the potentially interesting ski areas, we can use general highlighting techniques that will inform us about the location of these ski areas and their frequency. But again, we have to repetitively zoom in and out to compare their details, which isn't efficient. And to overcome this lack of navigational cues, one approach is to magnify these patterns that are too small to be perceivable and just show them as an inset, this kind of thumbnail here within the view. And while that works just fine for up to a few patterns, we of course quickly run into troubles as the number of potentially interesting patterns grows. And ideally, we would like to be able to see the visual details for all of these potentially interesting patterns here as an inset within the view. And every inset should ideally be as close to its origin as possible without overlapping other insets or other important visual patterns within that view. And now to make this approach scalable, we also somehow need to control how many insets we are showing simultaneously. And to address all these requirements, we have developed scalable insets, which is a novel technique that employs a dynamic trade-off between the number and size of insets, their locality, so how close they are to their origin, as well as how much they preserve the context. And so in scalable insets, each potentially interesting pattern that is too small to be identifiable at the current zoom level is displayed here as an inset within the view. And whenever two or more of these patterns are very close to each other, they get aggregated into these groups to save space. And the group here in this case shows up to 4 representative patterns in addition to the group size. And now these insets are placed such that they are as close to their origin as possible, while minimizing occlusion with other insets as well as other annotated patterns within the view. And depending on the importance of the context here, so the map in this case, these insets can either be placed on the outer boundary or directly within the view, which I'll show you in a second. But most importantly, every aspect here of scalable insets is dynamic. So meaning as you zoom into the map, the placement and also aggregation level of these insets is dynamically updated. And in general, as you zoom into a specific region, you'll see more details about a group of aggregated patterns. Now, scalable insets also works with other multi-scale visualizations. So here, for instance, on the left, we see an example of user annotations that are being highlighted using insets in a gigapixel image. And on the right, you see an example of highlighting these dot-like patterns in a genome interaction map. And on a more technical side, to realize this placement of insets, we've developed a simulated annealing-based algorithm that is run for a few cycles on every zoom event, with the objective to minimize the distance of insets to their origin, also minimize the movement per iteration, and the overlap with other insets and other annotations, as well as leader line crossing. For grouping insets, we've built an approach that is based on the distance of the inset's origin, so meaning the pattern annotation in pixel space. And to avoid abrupt changes, the distance threshold for merging or splitting these groups differs. Now, in order to actually find out whether scalable insets is really useful for exploring patterns in multi-scale visualizations, we conducted 2 user studies. In the first controlled user study with 18 participants on natural image data, we compared both of the scalable insets techniques, so the inner and the outer placement techniques, against a bounding box highlighting technique. On frequency estimation, so how many annotations are in a certain view, pattern comparison, and also pattern search. And overall, what we found at a high level is that scalable insets can make pattern search on average by up to 30% faster, and also improve the accuracy of comparing different pattern types by up to 20%, while adding relatively little overhead. So the frequency estimation of patterns is about 5% slower compared to bounding box highlighting. Now, in the second, more qualitative user study with 6 computational biologists, we compared the user behavior between the inner and the outer placement technique. And what we found is that when these insets are placed within the view, that encouraged more guided navigation. So the participants kind of spent more time zooming into an area that contained some insets with interesting patterns. On the other hand, when the insets were placed on the outer boundary, participants spent more time searching for patterns and comparing different patterns to each other, presumably because they are nicely aligned on the outer boundary. And so regarding our goal to improve navigating in these multi-scale visualizations, we can conclude that scalable insets provides an efficient pattern-driven guidance that can significantly reduce the time needed to find these small local patterns. And moreover, our technique can improve the discovery of unexpected patterns that would otherwise go unnoticed, simply because the search space or the exploration space is so large. And finally, on a technical level, we provide evidence that this kind of grouping that we've seen here in scalable insets is an effective approach to really ensure visual scalability. Now, the obvious next question is, I see a question coming in. So Chris asks if the jiggling of insets can be eliminated if the algorithm determines that there are no new insets be added during zoom and pan. So the during pan, maybe I should first of all say that there is, when we're panning, we're not recomputing the layout at all, except for new insets that come in. For zooming, that's not possible, at least currently with our simulated annealing-based approach, as for instance, the space that is covered by other patterns in the view changes. So the objective actually might change as you zoom in or out. So as you, for instance, zoom in, an existing pattern annotation might blow up. So you have to basically also push out these instances at the same time. But during pan, you're right, we don't have to update anything. So the obvious next question is, or maybe not super obvious, but one of the next questions is, like, if we have such a system like this, what if we want to compare patterns irrespective of their location? So right now, scalable insets is tailored towards helping you find regions of interest. But in certain cases, we might not matter, for instance, care whether, for instance, a pattern is up here or it is down here. And one approach to we could take is, for instance, cut out all of these instances and show them in some kind of small multiple setup. So meaning we plot them in a grid like this. And while this allows us to kind of compare neighbors easily, overall, this gets pretty overwhelming. And to address this challenge, we created a new approach called interactive piling, or we generalized this approach, which is useful for interactively organizing and comparing large number of these small multiples. And we published this work also at the IEEE InfoVis conference in 2020, where it received a best paper honorable mention. Now, in general, small multiples is a widely used technique to display large or multivariate data sets through a series of these miniature visualizations that can show either different facets, dimensions, or subsets of the data set. However, as we've just seen, like, a major challenge with small multiples is scalability. And it is, of course, not limited to genomics. So here, for instance, on the left side, we see a large collection of photos showing cars in context. In the middle, we see necklace sketches. And on the right, we have small multiples of area charts of COVID-19 infection rates for countries across the world. And now, as we kind of see, with more and more small multiples, comparison becomes less effective due to, for instance, the decreased availability of screen real estate per visualization. So we simply have, less pixels available to draw each individual photo in this case. Or we might have to deal with extensive navigation that is needed to actually inspect the details of particular small multiples. And in either case, we have to deal with increasing efforts for sequentially scanning different small multiples here. Moreover, questions regarding multiple items is kind of challenging. So for instance, estimating the cumulative infection rate of 3 or more countries is really challenging. Now, to address this challenge, we can employ interactive piling. And inspired by the act of physically piling paper documents like these books here, piling describes an interface for interactive spatial grouping of these small multiples into piles of partially overlapping items. And so visually, a pile consists of 3 types of elements. So we have these partially overlapping items that make up the pile. And then additionally, we have something called a cover item that is shown on top of the pile in its entirety. And in addition to that, we also have a set of preview items like these book spines here that can hint at the visual details of individual items. But how should we actually go about designing such piles, given that there are many possible pile encodings? And to help answer this question, we systematically analyzed previous piling interfaces from the human-computer interaction and visualization community to, first of all, identify common goals and tasks that apply across different use cases. And then based on this literature review, we defined the first generalized design space for interactive visual piling. And we structured this design space according to the following 5 high-level analytical tasks that we think any piling interface should support, which is grouping, arrangement, previewing, browsing, and aggregation. And so for grouping, we distinguish between the manual grouping, which is often important for personalized organization and comparison, to automated grouping, which is required for scalability. For arrangements, we consider the relative positioning of piles of items on a pile and the absolute positioning of piles to each other, which can encode additional information. For content awareness, piles can implement 5 different layout types to preview items. And the choice here really depends on the visualization type as well as the application-specific tasks. And now in the context of piling, we regard browsing as the act of inspecting the visual details of piled up items. And I'll show you what that means in a minute. And then finally, aggregation is the act of visually summarizing these piled up items into a more concise form for enhanced content awareness and also comparison. And now to enable others to design their own piling interfaces, we have developed a JavaScript-based library that we call piling.js. And piling.js is data agnostic and built around the declarative view specification to avoid having to write low-level code. So for instance, to get started, we only need to provide the data, a matching renderer, and a view specification, and then piling.js manages the view state for us and provides browsing interactions. And so for instance, piling.js implements common mouse gestures like drag and drop, lassoing, etc. But moreover, piling.js also offers means to automatically arrange and group piles based on their underlying data. So as we can see here in this small example. Now, to put this all together, I want to show you how piling enhances the exploration of necklace sketches from Google Quick Draw that we saw earlier. So these sketches here are coming from users around the world who had 20 seconds to hopefully draw a recognizable necklace. And our goal is to analyze this collection of sketches in order to find clusters of recurring necklace concepts. And so here in this case, we'll start by arranging these sketches according to a UMAP embedding to gain an overview of the entire data set. And the embedding here is placing similar sketches close to each other to highlight clusters within the data set. Now, to simplify this view, we can automatically pile up items that are co-located within the same grid cell and then further adjust the grouping manually. And then here in this case, as a pile summary, we are averaging the grouped sketches, which enables us to identify different concepts of the necklace. We can further compare these concepts, for instance, by looking at these piles in isolation, as for these two examples here. And then finally, visual piles also offer encoding related metadata. So for instance, here we're going to visualize the geographic distribution of users who drew these sketches, which will enable us to see that in Southeast Asia, an open beaded necklace is more commonly drawn than an open pendant necklace. And so one of the exciting things is that piling can be really used in many different use cases. So here are a couple more examples from, for instance, immunofluorescent microscopy, machine learning, geography, genomics, public health, and more. And if you like, you can browse these examples online. And so in conclusion, visual piling really provides an interactive trade-off between classic small multiples and static summary visualizations. And this makes it particularly useful for pattern-driven cluster and time series analysis by reducing the visual complexity. And overall, we hope that in combination, our design space and the JavaScript library provides a framework for others to really create new piling interfaces more efficiently for their use cases. All right, and before I go into the next project, let's see if we have a question. Linda is asking, why is this not just clustering? So I think the primary difference here is that it's user-driven clustering in some sense that is being defined interactively on the fly. So upfront, for instance, you might not know by which dimensions you want to cluster your data. And so you might want to check multiple different dimensions on the fly and see how these clusters actually look like. And so that's really where piling comes in, to give you basically the tools to drive this clustering on the fly. All right, and so coming to my last project, you might have noticed that so far I've always assumed that we either know where patterns are located or that we have some kind of algorithmic tool that finds or detects pattern candidates. But what if that is not true and all we have is some kind of image in our mind what we're looking for? And to address this, we've created Peaks, which is a system for interactive visual pattern search in sequential data that is using unsupervised deep representation learning. And this work was published at the Euroviz conference in 2020, where it received the Best Paper Award. And so in general, visually searching for patterns in sequential data can be challenging when the search space is large, the patterns are complex, or the query is difficult to formalize. And to overcome these challenges, we need an effective feature representation that hopefully captures the important visual properties for similarity comparison. And so for instance, in the case of this bar charts here, such features could be the number, height, or position of peaks, their magnitude, and so on. But typically, this feature representation is defined up front. And while this can be useful to find specific patterns, it requires a deep understanding of the data itself, and it often does not generalize well. And so to provide a more generic solution, we have developed a convolutional order encoder model for unsupervised feature learning. And the idea behind that was pretty simple. So we thought, like, if CNNs can detect patterns in natural images, maybe they can also detect patterns in abstract information visualizations. And so given a region of this sequential data, the first part of our model learns a latent representation that then the second part of the model uses to reconstruct the input data. And by having this lower dimensional latent representation, the autoencoder is kind of forced to learn an abstraction that hopefully captures important visual features of the data. Now, additionally, to support interactive visual pattern search, we not only need a good matching technique, but also a system that is able to adapt to the user's perceived similarity. So you can imagine sometimes maybe the positioning of peaks is really important, while in other cases, the shape of the pattern is more important, or the magnitude. And to address this, we have developed a system called Peaks that uses our novel representation learning technique and combines it with an active learning strategy to allow the user to interactively train a classifier in this feature space here that identifies the important visual dimensions using binary relevance feedback. And now the first question is, of course, can our deep representation learning technique really capture all of the salient properties? And so here we see 9 diverse examples from a DNA sequencing data set, where the original data is shown at the top in black. Then below that is the reconstruction in blue. And then the third instance here is a difference plot. Now, what we can see is that overall, the autoencoder provides a smooth reconstruction of the input data and is able to encode most of the salient visual features. But you might wonder, reconstruction wasn't the goal here, really. So the next question is, of course, is this learned encoding really useful for similarity search? And the answer, in our case at least, is yes. And so we compared our technique against a set of 6 other commonly used techniques for similarity search in sequential data, using these 9 diverse search queries that we show here at the top. And looking at some of these details here, we see that each of the other techniques can fail in certain cases to capture important visual aspects of the pattern, as highlighted here with these black boxes. And while no technique provides perfect results, our autoencoder model is able to capture more important visual aspects. And now, importantly, we were able to confirm these anecdotal results in 2 user studies on Amazon Mechanical Turk. Now let's take a look at the user interface to see how all these aspects actually play together. So at the top here, we have the query view, which is pan and zoomable and shows the initial query region that we're searching for. Below that is the list view that shows multiple regions for comparison and labeling. And then on the right, Peaks features an embedding view, which provides an overview of all the genomic regions and how they compare in terms of their similarity in the learned feature space. And finally, at the bottom right is the progress view, which shows properties of the interactively trained classifier to help the analyst determine when to stop the labeling process. And now in the following, I'll show you snippets from a live recording where we trained a classifier from scratch to find these slightly asymmetrical peaks in 2 genomic data sets. And these kind of asymmetrical peaks here can indicate the directionality of an actively transcribed gene start site. So there's like a biological reason for finding those. Now, in order to build a classifier, we need to specify a search query and then provide feedback to the system in form of these binary labels. And here in this case, we start labeling regions selected by our active learning sampling strategy as either matching our search target, not matching our search target, or being inconclusive by these clicks on the crossmark or checkmark button. And at the end of our initial sample set, we'll train our first classifier and then retrieve a new set of samples. And now as we continue labeling regions and training classifiers, the progress view here on the bottom right shows us the state of the learned classifier. So for instance, we can see that the uncertainty here gradually decreases. Now to gain an overview of all regions, we'll take a look at the embedding view. And in this view, neighborhoods of these blue dots indicate regions that kind of match our, or are predicted to match our search query. And now we could use this view to explore these neighborhoods in detail, but for now, let's turn our attention to the results to assess our classifier's performance. And besides just browsing these results, we can further improve our classifier by continuing to label regions to, for instance, confirm true positives and also flag false positives. And now eventually our labels might disagree with our classifier due to inconsistency, and Peaks notifies us about such conflicts. So for instance, here in this case, we are fixing incorrectly assigned positive labels, which we do not consider matches anymore. And then ultimately, once we're happy with the current results, we can explore the entire data set in the query view at the top. And to guide this exploration, this view features a light gray to dark blue heat map, where dark blue represents regions in the data that match our initial search query, as we see here, for example. Now, we evaluated the usability and usefulness of Peaks for building classifiers from scratch and also from existing labels. And through a user study with 8 computational biologists on simulated data, we first of all found that even though the system looks complex, a 15-minute introduction was typically sufficient to enable participants to successfully build and refine pattern classifiers. But more importantly, we also found that on average, the performance of the classifier was higher when they were trained on the expert labels versus training them on the ground truth labels that were sampled with our active learning strategy. So which really indicates that the expert steered training process, at least at the very beginning, can be more effective than a fully automated approach. And so in summary, with Peaks, we kind of provide an interactive pattern search approach that we see as a complement to specialized feature detectors. And we think it's particularly useful for quick hypothesis testing of not well-defined patterns, and to also increase the trust in the results of existing annotated patterns. Another lesson learned is that the generative models here are really useful for human AI interaction, as their output enables kind of interpreting the model state, at least to some extent. So for instance, by showing the reconstruction here in this, as these pink chart bars behind the actual data, an analyst can check, and if the specific aspects that they're searching for is not represented in this pink chart, then they might decide to just cancel the search before even starting to label. And finally, with this project, we also provide further evidence for the benefit of like human AI interplay, and show that it can be really effective to overcome issues like cold start problems, where the human really provides the expertise and AI brings in the scalability. And so let's zoom out again to the big picture. The 4 projects that I've shown you today really provide solutions for more efficient browsing, comparing, and finding patterns at and across scales. And research-wise, these projects contribute technical and empirical knowledge to the visualization community, but they also expand our understanding in interdisciplinary fields, like, for instance, biovisualization, and they provide new ideas for emerging areas like machine learning for visualization. And since I'm a huge fan of open source software, the code for all of these projects is freely and openly available on GitHub to really allow others to use these tools in their own applications. And overall, these projects are part of my broader mission towards more scalable human data interaction, and are accompanied by other work that I've significantly contributed to in, for instance, semantic data modeling and exploration, data compression, and also design feedback, which I'm happy to talk about offline. And now, of course, the work I've just shown you is just the first step. And so I think there are a number of open challenges that are really exciting avenues for future research. And so, for instance, composing these linked views for browsing multimodal and multi-scale data that I've shown you at the very beginning is currently an entirely manual process. And there isn't really any guidance on how to lay out these different views, and more importantly, what data or pattern annotations to load. And to address this, I'm planning to develop methods and tools for guided authoring of these composite figures by first of all analyzing how biologists, for instance, currently create these figures. And I also want to explore whether storytelling techniques that were, for instance, found to be successful for data comics can be used to support scientists in creating narratives as they explore their data for improved understanding. So for instance, as you can see here, the layout between like this composite figure and the data comic share many similarities. Also, I would like to explore adaptive guidance that is based on the user behavior to provide context-specific suggestions and maybe also even visual adaptations. Now, in terms of pattern exploration, this interactive piling approach I've shown you is still a relatively new technique. And we don't have a lot of empirical data for concrete recommendations regarding pile encodings and also the pile interactions. And to further improve the usefulness of interactive piling, I plan to formalize the design process by first of all developing a Vegalite compatible grammar for designing this pile interface, and then to evaluate the perceptual effectiveness of different pile designs, with the goal of really enabling automated reasoning for design recommendations. Complementary to this, I'm also interested in studying adaptive and intelligent interactions to improve the efficiency of piling in different use cases. And finally, I believe there's a great opportunity to generalize Peaks's pattern search approach to other, for instance, 2D and multimodal visualizations, given that we know that CNNs can, for instance, successfully learn to recognize patterns in other visualization types as well. And complementary to this, I started exploring ways of improving the visual labeling process, which I think is key to this approach, but is also, of course, a major bottleneck currently. And so, for instance, labeling multiple instances at once, propagating labels, or inferring maybe even label functions from visual feedback for integration into weak supervision approaches like Alex Ratner's Snorkel could greatly improve the label efficiency. And finally, I would also like to further enhance the human-AI interaction for more effective labeling and also to mitigate biases. So for instance, it's currently hard to evaluate as an analyst what the impact is of newly added labels on the next iteration of the interactively trained machine learning model. And to address these challenges, I want to develop and test new visualization approaches of the whole learning process, and maybe even add explanations, visual explanations to the impact of labels as well. And so with that, I'd like to thank you all for your attention, as well as all my wonderful collaborators and funding agencies who made all of these great projects possible. And I'm happy, of course, to take your questions now.
To hear the applause, so I'll add it. But yeah, thank you, Fritz. And so I encourage others to share questions either in the Zoom chat or out loud if it doesn't get too cacophonous. I see Tim has a question, and others come in. I'll just start with one for you, Fritz, which is you showed a lot of great examples of a smooth interaction and animation actually as part of the design of your many systems. And I wondered if you had, alongside the overall design principles, rendering or data management insights that you also needed to apply to arrive at those application results.
Yeah, so one of the, maybe in terms of the data management aspect, one thing I haven't explicitly talked about is, so the underlying foundation of most of these systems is HiGlass. And in order to get this scalable, basically, we've developed a a so-called tileset API that can be overlaid on top of the data that provides access to just the chunks that are needed for being rendered. And I guess the key important thing there was that it's like a separate API that can be added on top of any others, domain-specific data storage solutions. So it makes it more easy to integrate new data sets into the whole system. Yeah, on the rendering part, so I guess the trickiest thing is always like, for instance, for piling, we had to always revert back to WebGL simply because like SVG solutions don't scale. And our current approach is basically to determine upfront where do we need interaction, and when can we give up certain interactivity for more speed. So for instance, in piling.js, you take an SVG image, you render it into a texture object, and then continue to working on this texture object instead of saying like, okay, we need the interactivity for every single element that we have in that view. That can be added back later, basically, once we want to explore a specific instance in detail. So Tim is asking about the reconstruction quality, which wasn't great for some of the examples I've shown you. And yeah, that is true. In some cases, the reconstruction isn't perfect and some of the peaks can be missed, but so there is a more fundamental question of, do we really need perfect reconstruction in a search system, like in a visual search system? So for instance, I had the same, sometimes closely located peaks are basically being merged together. And I showed that to the biologists and was like, oh, that might not look good. And they were like super happy with that because they figured that, due to high frequency noise, it's actually great if you smooth out certain peaks that are closely located. In order how to better address this, I mean, a simple approach is to lower the size of the regions that you're looking at all at once, essentially. So as you make those windows smaller, or as you zoom in more, you get better accuracy. But you lose accuracy as you make this genomic views essentially larger because the pattern frequency sort of increases. I hope that answers the question, at least to some extent. So Paul is asking, how important are the 1D and 2D aspects of these applications for the success? Well, I think it depends on, so some techniques are specifically made just for 1D use and others are specifically made for 2D use. So certain techniques will not work in one case or the other. So for instance, the peaks currently doesn't work for 2D data, but there's nothing that prevents us from extending that out and providing a different structure, like network model, to capture other data types. Specifically depends on the task that you're trying to solve. For instance, scalable insets can also be applied to one-dimensional data. There isn't any fundamental issue that prevents us from applying it to that system. And in that case, we have only applied it to two-dimensional data because that's where we currently saw a lack of techniques to help us really navigate these data. All right, Sheng Long is asking, yeah, so what is the magic behind? I think I've answered that with the Jeff question to some extent. So basically, for to visualize these systems, we only request data that is tiled in a sense. So we only get a certain resolution of the data back that is basically tailored to some extent to the view, so that we're not requesting too much data. The interactivity, we remain by not sending pre-rendered data. So the data might be pre-aggregated, but it is never pre-rendered. So that allows us to, on the fly, for instance, change the visual encoding without having to continuously request new data.
with that, let's thank Fritz again. Thank you, Fritz, for joining us today. And thank you all for joining the talk.